Cancer is one of the biggest medical problems of the 21st century. According to the CDC, it is the second leading cause of death in the United States. Many different treatments exist, ranging from chemotherapy to surgery. One of the newer treatments for cancer is immunotherapy.
Immunotherapy utilizes the patient's own immune system to fight off disease. Usually, the disease we're trying to fight off with immunotherapy is not fought off by the patient's immune system without technological interference. CAR T cells are a form of immunotherapy in which genetically engineered T cells hunt down and attack cancer cells. The CAR T cells have receptors on their membranes which can bind to antigens on the surface of their target cell.
To get cells to produce these receptors, they are genetically modified using viral vectors. Viral vectors are viruses that are stripped of their ability to cause damage to cells, but keep the ability to integrate with a cell's genetic information.
The new characteristics of the T cells are then expressed after the newly inserted DNA is transcribed and translated into proteins for the cell. First, DNA is split into two separate strands, called the sense and antisense strand. The sense strand is copied into mRNA when the enzyme RNA polymerase transcribes the antisense strand into mRNA. This mRNA has caps added to the 5' and 3' ends. The new mRNA strand leaves the nucleus and enters the cytoplasm. A ribosome binds to the mRNA strand in two parts. Then the ribosomes again to match anticodons carrying amino acids to codons on the mRNA strand. The amino acids form a chain and then become a protein.
This is what a normal T cell looks like. This is what a T cell looks like after the newly introduced DNA has been expressed. The new parts are circled in red. Using the new DNA from the virus, the T cell acquires new parts which turn the T cell into a CAR T cell. These CAR T cells now have monoclonal antibodies from the DNA, which are called single-chain variable fragments, or SCFVs. These SCFVs on the outside of the T cell's membrane are linked to stimulatory molecules on the inside of the cell, which were also added by the newly inserted DNA.
What the single-chain variable fragment does is guides the T cell to its target antigen, which is mostly cancer cells, so that the cell can bind to its target. Once the cell binds to its target, the stimulatory molecules in the T cells, Stim1 and Stim2, provide signals for the T cells to become fully active, which allows the cell to effectively attack and kill cancer cells.
This is a picture of a CAR T cell attacking a cancer cell. The cancer cell is shown in blue, and the CAR T cell is shown in green. Here are the results from a clinical trial done using CAR T cells. On top are pictures of the cancer before the CAR T cells were introduced, and on the bottom you can see the cancer afterwards. The tumor has greatly shrunk in size, showing that CAR T cells can be a viable treatment for cancer.
The process of creating the CAR T cell is very similar to the Piglo lab we performed in class. In both, we introduced new DNA to get the cell to produce new proteins, such as the GFP, or green fluorescent protein, we made the bacteria express in the Piglo lab.
To introduce the new DNA in the bacteria, we used plasmids, small rings of DNA, which a bacteria like E. coli can use to produce proteins. Both the lab and the CAR T cell involve inserting new DNA into a cell to get it to produce a new protein, but they differ because the T cell uses viral vectors to insert the new DNA, and the Piglo experiment uses a plasmid to insert the new DNA.
The reason we used a plasmid to insert new DNA for the bacterial cell is because it was a prokaryotic cell, which means it has no nucleus protecting its genetic code. This means we just had to insert the DNA into the bacteria's cytoplasm to make new proteins form in the bacteria. The T cell, however, was a eukaryotic cell, which means it has a nucleus protecting its genetic code. This means if we just inserted the DNA into the cytoplasm, the DNA would do nothing. Instead, we had to use a viral vector, as viruses have the ability to bypass the nucleus of the cell and integrate itself into the cell's DNA.
CAR T cells are a revolutionary new cancer treatment that engineers T cells to hunt down and kill cancer cells in the human body.